The LYSOCIL project addresses a scientific strategy to reinforce research and innovation excellence in the study of diseases that affect cilia, with special attention to the diagnosis and understanding of the causes of these diseases. Cilia are finger-like projections that exist in most of the cell types in our body. There are two types of cilia, motile and non-motile. Motile cilia move fluids in our body, such as mucus in the respiratory airways, where dust particles and microorganisms become trapped. Therefore, they work as brooms to clean the airways. On the other hand, non-motile cilia work as antennas, receiving signals from the extracellular environment and relaying them to the cell's interior. For example, in the kidney tubules, cilia sense the urine flow by bending and transmitting a mechanical signal to those cells. Diseases that result from failure in ciliary functions are collectively named ciliopathies. The symptoms range from obesity to mental retardation, developmental defects, and kidney cysts. Moreover, the severity of these diseases can also range from mild to severe, which can be lethal early in life. At CEDOC, the Chronic Diseases Research Center of Nova Medical School, we investigate how cilia are formed and maintained, and the mechanisms of ciliopathies, namely primary ciliary dyskinesia. This rare disease affects 1 in 10,000 newborns and has caused mutations in more than 40 different genes. Primary ciliary dyskinesia causes recurrent respiratory infections due to the accumulation of mucus in the airways, where microorganisms can proliferate, as well as the misplacement of the organs in the body. To study these questions, LYSOCIL and CEDOC researchers used ciliated cells from human nasal epithelia and zebrafish as an animal model. Zebrafish is an excellent model to study ciliopathies since it has many ciliated tissues with motile and non-motile cilia, single or multiciliated, and its transparency allows scientists to observe live processes in real time. By mimicking human mutations in zebrafish, LYSOCIL scientists are currently testing new therapeutic approaches to restore motile cilia function in zebrafish larvae that recapitulate the ones from patients with primary ciliary dyskinesia. At CEDOC, researchers have managed to rescue cilia motility in the nose of the fish larvae. These larvae had immotile cilia, whose motility was restored by providing the correct gene that was missing. LYSOCIL, a new alliance in knowledge and research for a new hope in rare diseases.